In this video, we will discuss about the pathways that is suppressed by TXA2 that will prevent T cell activations. So, in order to find out what are the pathways that get affected by this TXA2, what it did is they did this RNA sequencing analysis and compare it with different conditions. That is, in the absence of TXA2 analog at wild type and this ARHGF1 knockout, and in the presence of TXA2 analog and the wild type and ARHGF1 knockout. What they are able to see is that genes that are responsible for this genes that are responsible for the migrations are highly upregulated in the wild type cells, but they are downregulated in the conditions where this uh, cells are treated with TXA2 analog, but they are not get affected in the case where ARHGF1 has been knocked down, indicating that the ARHGF1 plays a role towards this T cells activations. So T cells activation genes are down regulated where in the wild type cells which is affected with TXA2 analog. And these things they have also validated using the GSE analysis of this naive cells, but this memory CD8 plus T cells. They are able to see that the genes are highly upregulated in the naive T cells, but they are downregulated in this CD8 plus T cells in the conditions of this wild type, which is treated with this TXA2 analog, that is preventing the T cells activations. And thereafter, what they did is they look into the literature and find out that this survival pathways gets affected by whenever these cells get treated with this anti-CD3 cross-linking. So they check this phenomena in the flow cytometry, and here we can see that in the case of no stimulations they do not see too much this PERK signaling pathways or phosphorylation of this S6 or AKT pathways, both in the case of wild type and ARGF1 knockout. But when they stimulate it with this anti-CD3 cross-linking, they can see the upregulations of this both PERK phosphorylation pathways and this phosphorylation of this S6 in the case of wild type cells, and also in the case of ARHGF1 cells. But when they treat it with TXA2 analog, they are able to see the suppressions in the case of these wild type cells, but not in the case of ARHGF1 knockout, indicating that this ARHGF1 plays a role towards this PERK phosphorylations pathways and these activations of AKT also, or ERK and AKT pathways. And this is the reasons when the TXA2 analog is used, it will trigger this ARHGF1 to down-regulate this PERK AKT pathways. And this PERK AKT pathways is dependent on this Rho or ROC signaling pathways. Downstream of this PERK signaling pathways or ARHGF1 will interact with this Rho signaling pathways. So they also check this Rho GTP presence in the TXA2 analog stimulated cells, and they can see that in the wild type cells there is upregulations of these Rho signaling pathways that will cause the suppressions of this ERK and AKT pathways. We can see that when the Rho signaling pathways is increased, the phosphorylations of this AKT goes down. But in the case of this knockout, ARHGF1 knockout, there is no Rho signaling pathways that are activated. So there is not much effect, or there is no effect on this phosphorylation of the AKT pathways. And these things they validated using the flow cytometry also studies also. What they did is they artificially inject this activated row in the knockout cell to see whether it will affect the phosphorylation of the AKT or not. And they did their studies by stimulating the cells with anti-CD3. So when there is no row is introduced, they are able to see around 14.3% of the cells that are ERK and AKT phosphorylations at S6 positives in the wild type, and in the ARHGF1 this number is high, that is 39.9%. But when they artificially introduce row CA, they are able to see suppressions not only in the knockout, because here the row is artificially introduced, so it can do its function to suppress peak phosphorylations of AKT, but it also suppresses this phosphorylation, this PERK and PSC positive cells, because of the overexpressions of this Rho A in the wild type. That can be seen here from 14.3 to 7.7 and 29.9 to 16.6. Then they check using the confocal microscopy that uh, they check that as soon as these TXA2 analogs are introduced in the wild type cells, they are able to see that it will activate these row signaling pathways that can prevent this phosphorylation of the AKT. So this triggering of this row signaling pathways is being shown here by the arrows in the TXA2 analog wild type cells. But these things are not so common in the case of ARHGF1 knockout. These things they have validated using the confocal that row interactions with this row interactions in the TXA2 analog wild type cells. And then they check the ratio of this row and P10. The row will finally affect these P10 pathways or P10 signalings. So then they check this ratio of P10 or the presence of P10, and they are able to see that when they use the TP inhibitors or ROCK inhibitors, there is a down regulations of this P10 uh, in the case of wild type conditions, but not in the case of ARHGF1 conditions. So they at this point hypothesize that it's the TP inhibitors or ROCK inhibitors that will affect these phosphorylations of the AKT and phosphorylation of the S6, phosphorylation of the AKT and phosphorylation of the this ERK pathways. So they did their Western blot analysis, and in this case, what they are able to find out is that for this phosphorylation of the AKT, it's dependent on this TP inhibitors, it's dependent on the TP, ARHGF1, uh, TXA2 analog, and CD3 cross-linking. As we can see that when all these three things are present, they are able to see upregulations of the phosphorylations of the AKT. But when they check this phosphorylation of the ERK, uh, it does not depend too much on this row. In the case of row also we see an increase in the phosphorylation of the AKT and the row inhibitors are used, but when we check for this ERK, we can see an increase when we use the TP inhibitors CD3 and TXA2, but there is not change has been observed when they use the ROCK inhibitors. So this phosphorylation of the ERK depends on TP, CD3 and TXA2, whereas phosphorylation of the AKT depends on TP, CD3, TXA2 and also the ROCK and P10. And all these things are almost remains common or uniform in the ARHGF1 knockout. So the things that they are able to find out in the panel G, that is TP inhibitors and ROCK inhibitors roles, TP and ROCK roles, and this phosphorylation of the AKT and PERK phosphorylation of the ERK pathways, and they check this using the Western blot where they activate in the case of no stimulations, they signals in the background, but when they stimulate the cells using anti-CD3, they can see in the wild type the phosphorylations of this AKT is increased. It can be seen here in the knockout, ARGF1 knockout. But when they use the anti-AKT pathways, they are able to see suppressions in these phosphorylations of this AKT. So this indicates that uh, it's the AKT, because of the AKT pathways they are able to, because of this AKT pathways they are able to see phosphorylations of this S6. And when they did their same studies in the, when they checked the cytokines, one of the cytokines, they are able to see the similar results. That is, when they use the AKT inhibitors, the cytokines production is also goes down compared to the vehicle, where cytokine production is around 68.8%, whereas in the case of AKT inhibitors, the cytokines levels goes down to 16.6, which is close to the background. And in the panel edge, the phosphorylations of the S6 goes down when they use the AKT inhibitors, indicating that it's the AKT pathways that plays a role, and the phosphorylation of the AKT does, AKT and ERK pathways just plays a role towards this ARGF1 plays a role towards this phosphorylations of this AKT, and the other pathways does not. There is no other, or the contributions by the other pathways is minimum. This is what they have finally concluded this panel H. So overall, they are able to conclude that it's the TXA2 suppresses TCR driven kinase pathways using this phosphorylation of the AKT and ERK pathways through this Rho and P10 signalings in the downstream of the ARHGF1 activated, the downstream of the ARHGF1 pathways. So I hope you understood to some extent how these signaling pathways get affected in the TXA2 analog, or in the TXA2 is presence, or the T cells get affected by the TXA2. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.